In this lecture, we will discuss about unfolded protein response. We will discuss about the statistics of GSEA in next lecture because we did not get time. I did not get time to prepare for that. So at this time, we will discuss about unfolded protein response. It is popularly known as UPR response. Okay. So when this is occurred, as we have learned in our previous videos, that it is essential for a protein to have its native structure to perform its function for which it is been secreted, for which it has been formed inside the cell. So if the protein is not been able to form its structure, its native structure, then it might lead to several stresses. It might lead to several different responses in the cell, and one of the the organelle in which this organelle of the cell in which this response originates is endoplasmic reticulum. If endoplasmic reticulum, and because of this unfolded protein response, it will lead to ER stress. This ER stress, and this will lead to unfolded protein response, and unfolded protein response has a different component. It is mainly divided into three parts. One is known as IRE1 alpha response, second is ATF6 response, and the third one is ATF4 response. In IRE1 alpha response, what happens is that in the unfolded protein, unfolded protein is that now the stress is on the endoplasmic reticulum because it is too much crowded with the unfolded protein. So the main thing the cell will do is to re-establish to its original position. For this, the things that cells will do is first decrease the translation of the generic proteins that does not have that much important role in the cell existence. So for this it will in order to reduce the translation of the unfolded protein response, it needs these three components together to take the role of this IRE alpha 1. In short, what it will do is there is a protein called XPB1. This should be the name, XPB1, and it is originally present as unspliced form, and with the ER stress because of unfolded protein response, this secretion of this protein will convert this unspliced form XPB1 to XPB as spliced form, and what it will does is it will now regulate the genes regulate genes that is responsible for ERAD and the chaperones. So how this will help in reducing the ER stress is that ERAD. What ERAD does is ERAD does to transfer the unfolded proteins towards the ubiquitination, that is producible degradation. And if we increase this ERAD, then it will try to reduce the amount of crowd inside the endoplasmic reticulum. At the same time, the chaperones. What chaperone does is chaperones helps proteins to get its native structure. If we increase the amount of chaperones in the endoplasmic reticulum, it will help the proteins to get its native structure. So both of these get regulated in the ER stress. And what is ATF4 is does? ATF4 regulates the XPB1 upregulation. So once this is upregulated, then its function will be taken care of by IRE1 alpha. And what this ATF6 does? ATF6 helps in regulating general proteins. So what it does is the proteins that are not necessary at this moment of time, it will try to reduce the translation of those proteins. So in that case, the amount of proteins that will come inside the endoplasmic reticulum will reduce. So we are acting on the three ways to reduce the ER stress. First, we are trying to make the native structure. We are introducing the chaperones, more chaperones, so that amount of unfolded proteins reduces. So unfolded protein will reduce because we are increasing the chaperones. And at the same time, we are increasing the proteasomal degradation, ERAD mechanism. Ubiquitination, we are increasing the ubiquitination via ERAD mechanism. So this will also help in reduce ER stress. At the same time, we are decreasing the input. The input is also reduced as we are not inputting any general proteins that is not required at this unusual situation, that is in ER stress. So I hope you will get an overview of what this unfolded protein response means. So we will cover our topic, that is statistics of the GSEA, in the next lecture. Okay, thank you.